Right now, welcome back. We're on a continuation of the former lecture. The specific topic of concern now is would be chaperones are proteins that prevent folding and unproductive interactions of other proteins. So this topic is part of the main subject of cell organization, which comes under this topic of what you call the proposed role of cargo carrying of NLS signals and all that, which comes as part of the importance and exportants that are involved in transport of macromolecules in and out of the nucleus, which comes under the mitochondrion that imports and synthesizes proteins, right? So we're going back to this portion and into the major details of it. We have portions that are continuation as well. So I'm just repeating this small para to give you the continuation, right? So exit from the ER may be the rate limiting step in the secretory pathway. In this context, it has been found that certain proteins play a role in the assembly or proper folding of other proteins, without themselves being components of the latter. Such proteins are called molecular chaperones. A number of important properties of these proteins are listed in Table 5, and the names of some of particular importance in the ER, and they're listed in the table below, right? So, some properties of chaperone proteins are they're present in a wide range of species, from bacteria to humans. Many are so-called heat shock proteins, HSP. Some are inducible by conditions that cause unfolding of newly synthesized proteins, example, elevated temperature and various chemicals. They bind to predominantly hydrophobic regions of unfolded and aggregated proteins. They act in a path as a quality control of editing mechanism for detecting misfolded or otherwise defective proteins. Most chaperones show associated ATPase activity, with ATP or ADP being involved in the protein-chaperone interaction, found in various cellular compartments such as cytosol, mitochondria, and the lumen of the endoplasmic reticulum. Now, if you see this word HSP, heat shock protein, this could be an easy MCQ question that comes with exams. What exactly does HSP stand for? What exactly are heat shock proteins? So that's the way you reason out. So once I finish this topic, there'll be Q&As on this. Likewise, Q&As for the former. Okay, this would be at the end of each month. So we'll spend 1 or 2 days on Q&As, right? So before that, you get to know the details of the subject matter dealt with. So there are about 13 topics, I understand, for the CSIR-NET syllabus of life sciences. So let's finish all this page by page in detail, so you get to have a clear understanding of everything. And don't forget to send me your queries on the comment section, right? I'll be around. And you could always connect with me on my email or WhatsApp too. You do have my email ID, I guess. Well, I just repeat it once again for you. It's called newagedentalrehab@gmail.com. Or you could go for my alternate email, which is drjacob_bk@yahoo.com, right? Okay, continuing with the next portion. The next table will be some chaperones and enzymes involved in folding that are located in the rough endoplasmic reticulum. You have BIP, which is immunoglobulin, heavy chain binding protein, GRP94, which is glucose regulated protein, calnexin, calreticulin, PDI protein disulfide isomerase, PPI peptidyl, prolyl cis trans isomerase, right? Basically, they stabilize unfolded or partially folded intermediates, allowing them time to fold properly and prevent inappropriate interactions, thus combating the formation of non-functional structures. Most chaperones exhibit ATPase activity and bind ADP and ATP. This activity is important for their effect on folding. The ATP chaperone complex often has a high affinity for the unfolded protein, which when bound stimulates release of ADP with a replacement by ATP. The ATP chaperone complex in turn releases segments of the protein that are folded properly, and the cycle involving ADP and ATP binding is repeated until the folded protein is released. Right? Several examples of chaperones were introduced above when the sorting of mitochondrial proteins was discussed. The immunoglobulin heavy chain binding protein, that's BIP, is located in the lumen of the ER. This protein will bind abnormally folded immunoglobulin heavy chains and certain other proteins and prevent them from leaving the ER, in which they are degraded. Another important chaperone is calnexin, a calcium binding protein located in the ER membrane. This protein binds a wide variety of proteins, including mixed histocompatibility MHC antigens and a variety of serum proteins. Calnexin binds the monoglycosylated species of glycoproteins that occur during processing of glycoproteins, retaining them in the ER until the glycoprotein has folded properly. Calreticulin, which is also a calcium binding protein, has properties similar to those of calnexin. It is not membrane bound. Chaperones are not the only proteins in the ER lumen that are concerned with proper folding of proteins. Two enzymes are present that play an active role in folding. Protein disulfide isomerase, PDI, promotes rapid reshuffling of disulfide bonds until the correct set is achieved. Peptidyl prolyl isomerase accelerates folding of proline-containing proteins by catalyzing the cis-trans isomerization of X-pro bonds, where X is any amino acid residue. Transport vesicles are key players in intracellular protein traffic. Just before I continue on that, let's just see if we've left out anything below. No, that's a good continuation there. Right, so most proteins that are synthesized on membrane-bound polyribosomes and are destined for the Golgi apparatus or plasma membrane reach these sites inside transport plasma membrane, inside transport vesicles. The precise mechanisms by which proteins synthesized in the rough ER endoplasmic reticulum are inserted into these vesicles are not known. However, those involved in transport from the ER to the Golgi apparatus and vice versa, and from the Golgi to the plasma membrane, are mainly clathrin-free, unlike the coated vesicles involved in endocytosis. For the sake of clarity, the non-clathrin coated vesicles will be referred to in the text as transport vesicles. Right? There is evidence that proteins destined for the membranes of the Golgi apparatus contain specific signal sequences. On the other hand, most proteins destined for the plasma membrane or for secretions do not appear to contain specific signals reaching these destinations by default. The Golgi apparatus is involved in glycosylation and sorting of proteins. The Golgi apparatus plays two important roles in membrane synthesis. First, it is involved in the processing of the oligosaccharide chains of membrane and other aniline glycoprotein, and also contains enzymes involved in O-glycosylation. Second, it is involved in the sorting of various proteins prior to their delivery to the appropriate intracellular destinations. All parts of the Golgi body or the Golgi apparatus participate in the first role, whereas the trans-Golgi is particularly involved in the second and is very rich in vesicles. Because of their central role in protein transport, considerable research has been conducted in recent years concerning the formation and fate of transport vesicles. Model of non-clathrin coated vesicles involves SNAR and other factors. Vesicles lie at the heart of intracellular transport of many proteins. Recently, significant progress has been made in understanding the events involved in vesicle formation and transport. This has transpired because of the use of a number of approaches. These include establishment of cell-free systems with which to study vesicle formation. For instance, it is possible to observe by electron microscopy budding of vesicles from Golgi preparations incubated with cytosol and ATP. The development of generic approaches for studying vesicles in yeast has also been crucial. The picture is complex with its own nomenclature and involves a variety of cytosol and membrane proteins, GTP, ATP, and accessory factors. We're on to the table that shows the factors involved in the formation of non-clathrin coated vesicles and their transport. We have ARF, which is about ADP ribosylation factor. GTPase, COTOMA, a family of at least 7 coated proteins, alpha, beta, gamma, delta, epsilon, beta 1, and the last would be, I'll have to figure that out. I'll leave that for you to figure out, right? And different transport vesicles have different complements of coat proteins. SNAP would be soluble NSF attachment factor. SNAR would be SNAP receptor. V-SNAR would be vesicle SNAR. T-SNAR would be a target SNAR. GTP alpha S would be a non-hydrolyzable analog of GTP used to test the involvement of GTP. NEM, that's NEM, would be N-ethylmalamide, a chemical that alkylates sulfur hydryl groups, NSF, NEM sensitive factor, and ATPase. RAB proteins, a family of RAS-related proteins first observed in rat brain. They are GTPases and active when GTP is found. And NSF is a NEM sensitive factor and ATPase. Now, SEC1 is a member of a family of proteins that attach to T-SNARs and are displaced from them by RAB proteins, thereby allowing V-SNAR, T-SNAR interactions to occur. Now, based largely on a proposal by Rothman and colleagues, anti-retrograde vesicular transport can be considered to occur in 8 steps, as shown in figure 7. The basic concept is that each transport vesicle bears a unique address marker consisting of 1 or more V-SNAR proteins, while each target membrane bears 1 or more complementary T-SNAR proteins, with which the former interact specifically. Step 1, coat assembly is initiated when ARF is activated by binding GTP. So what's ARF? It's ADP ribosylation factor, right? ARF, right? And the basic concept is that each transport vesicle bears a unique address marker consisting of 1 or more V-SNAR proteins, while each target membrane bears 1 or more complementary T-SNAR proteins with which the former interacts specifically. So step 1 will be coat assembly is initiated when ARF is activated by binding GTP, which is exchanged for GDP. This leads to the association of GTP-bound ARF with its putative receptor hatched in figure 7 in the donor membrane. Step 2 would be membrane-associated ARF recruits the coat proteins that comprise the COTOMA shell from the cytosol, forming a coated bud. Step 3 would be the bud pinches off in a process involving acyl-CoA and probably ATP to complete the formation of the coated vesicle. Step 4, coat disassembly involving association of ARF and COTOMA shell follows hydrolysis of bound GDP. Uncoating is necessary for fusion to occur. Step 5 is vesicle targeting is achieved via members of a family of integral proteins termed V-SNAR that tag the vesicle during its budding. V-SNAR pair with cognate T-SNARs in the target membrane to dock the vesicle. It is presumed that steps 4 and 5 are closely coupled and that steps 4 may follow step 5, with the ARF and the COTOMA shell rapidly dissociating after docking. Step 6 will be the general fusion machinery that assembles on the paired SNAR complex. It includes an ATPase, NSF, NEM sensitive factor, and the SNAP, soluble NSF attachment factor proteins. SNAPs bind to the SNAP factor receptor complement, enabling NSF to bind. Step 7, hydrolysis of ATP by NSF is essential for fusion, a process that can be inhibited by an NEM and ethylmalamide. Certain other proteins and calcium are also required. Step 8, retrograde transport occurs to restart the cycle. This last step may retrieve certain proteins or recycle. V-SNAR, nicotazole, a microtubule disrupting agent, inhibits this step. Now, figure 7 is about the model of the steps in a round of anti-retrograde vesicular transport. The cycle starts in the bottom left hand here side of the figure, where two molecules of AR are represented as small ovals containing GDP. You see that, right? And the steps in the cycle are described in the text. Most of the abbreviations used are explained in Table 7. Okay. The roles of RAB and SEC1 proteins, see the text please, in the overall process and are not dealt with in this figure. CGN, cis-Golgi network, BFA, it's Briffeldin A. Now, Briffeldin A inhibits the coating process. The following points expand the clarity of the above-mentioned figure. 1. To participate in step 1, ARF must first be modified by addition of myristic acid, C14H20, employing myristoyl CoA as the acyl donor. Myristylation is one of a number of enzyme-catalyzed post-translational modifications involving addition of certain lipids to specific residues of proteins that facilitate the binding of proteins to the systolic surface of membranes or vesicles. Others are additions of palmitate, farnesyl, and geranyl. The two latter molecules are polyisoprenoids containing 15 and 20 carbon atoms respectively. At least 3 different types of KORAH vesicles have been distinguished: COPI, COPII, or shall I say COP1 and COP2, and clathrin-coated vesicles. The first two are referred to here as transport vesicles. Many other types of vesicles no doubt remain to be discovered. COP1 vesicles are involved in bidirectional transport from the ER to the Golgi and in the reverse direction, whereas COP2 vesicles are involved mainly in transport in the former direction. Clathrin containing vesicles are involved in transport from the trans-Golgi network to pre-lysosomes and from the plasma membrane to endosomes, respectively. Regarding the selection of cargo molecules by vesicles, this appears to be primarily a function of the coat proteins of vesicles. Cargo molecules may interact with coat proteins that either directly or via intermediate proteins that attach to coat proteins, and they then become enclosed in the appropriate vesicles. The fungal metabolite Briffeldin A prevents GDP from binding to ARF in step 1 and thus inhibits the anti-coating process. In its presence, the Golgi apparatus appears to disintegrate. The fragments are lost. It may do this by inhibiting the guanidine nucleotide exchange involved in step 1. And D would be GTP gamma S, a non-hydrolyzable analog of GTP, often used in investigations of the role of GTP in biochemical processes. Blocks disassembly of the coat from coated vesicles, leading to a buildup of coated vesicles. E, a family of RAS-like proteins called the RAP protein family are required in several steps of intracellular protein transport, regulated secretion, and endocytosis. The small monomeric GTPases that attach to the cytosolic faces of membranes via geranyl change. They attach in the GDP bound state, which is not shown in the figure, to the budding vesicle. Another family of proteins, SEC1, binds to D snares and prevents interaction with them and their complementary V snares. When a vesicle interacts with its target membrane, RAP proteins display SEC1 proteins and V snare. T snare interaction is free to occur. It appears that the RAB and SEC1 families of proteins regulate the speed of vesicle formation. Opposing each other, RAB proteins have been likened to throttles and SEC1 proteins to dampers on the overall process of vesicle formation. F. Studies using B snare and T snare proteins reconstituted into separate lipid bilayer vesicles have indicated that they form snap-ins, that is, snare complexes that link two membranes vesicles. SNAPs and NSM are required for formation of snap-ins. Once they have formed, they can apparently lead to spontaneous fusion of membranes at physiologic temperature, suggesting that they are the minimal machinery required for membrane fusion. G. The fusion of synaptic vesicles with the plasma membrane neurons involves a series of events similar to that described above. For example, one B snare is designated synaptobrevin, and two T snares are designated syntaxin and SNAP25, synaptosome-associated protein of 25-KDA. Botulinum B toxin is one of the most lethal toxins known and the most serious cause of food poisoning. One component of this toxin is a protease that appears to claim only synaptobrevin, thus inhibiting release of acetylcholine at the neuromuscular junction and possibly proving fatal depending on the dose taken. Although the above model describes non-clathrin-coated vesicles, it appears likely that many of the events described above apply, at least in principle, to clathrin-coated vesicles. I'm going on to the next topic. The assembly of membranes is complex. There are many cellular members, each with its own specific features. No satisfactory scheme describing the assembly of any one of these membranes is presently available. However, various proteins are initially inserted. How they're inserted into the membrane of the ER has been discussed above. The transporter proteins, including membrane proteins to various parts of the cell inside vesicles, has been described as well. Some general points about membrane assembly retain to be addressed. Asymmetry of both proteins and lipids is maintained during membrane assembly. Now, vesicles formed from membranes of the ER and Golgi apparatus, either naturally or pinched off by homogenization, exhibits transverse asymmetries of both lipid and proteins. These asymmetries are maintained during fusion of transport vesicles with the plasma membrane. The inside of the vesicles after fusion become the outside of the plasma membrane, and the cytoplasmic side of the vesicles remains the cytoplasmic side of the membrane, figure 8. Since the transverse asymmetry of the membranes already exists in the vesicles of the ER, well before they are fused to the plasma membrane, a major problem of membrane assembly becomes understanding how the integral proteins are inserted into the lipid bilayer of the ER. Note these diagrams down, take a copy of them and make your notes. Fusion of a vesicle with the plasma membrane preserves the orientation of any integral proteins embedded in the vesicle bilayer. Initially, the amino terminal of the protein faces the lumen or inner cavity of such a vesicle. After fusion, the amino terminal is on the exterior of the plasma membrane. That the orientation of the protein has not been reversed and can be perceived by noting that the other end of the molecule, the carboxyl terminal, is always immersed in the cytoplasm. The lumen of a vesicle and the outside of the cell are topologically equivalent. Phospholipids are the major class of lipid in membranes. The enzyme responsible for the synthesis of phospholipids reside in the cytoplasmic surface of the cisternae of the endoplasmic reticulum. As phospholipids are synthesized at that site, they probably self-assemble into thermodynamically stable bimolecular layers and thereby expanding the membrane and perhaps promoting the detachment of so-called lipid vesicles, right? And travel to other sites, donating the lipids to other members. However, little is known about this matter. As indicated above, cytosolic proteins that take up phospholipids from one membrane and release them to another, that is phospholipid exchange proteins, have been demonstrated. They probably play a role in contributing to the specific lipid composition of various membranes. Lipids and proteins undergo turnover at different rates in different membranes. It has been shown that half-lives of the lipids of the ER membranes of rat liver are generally shorter than those of its proteins, so that the turnover rates of lipids and proteins are independent. Indeed, different lipids have been found to have different half-lives. Furthermore, the half-lives of proteins of these membranes vary quite widely, some exhibiting short hours and others long days half-lives. Thus, individual lipids and proteins of the ER membranes appear to be inserted into it relatively independently, and this is the case for many other membranes. The biogenesis of membranes is thus a complex process about which much remains to be learned. One indication of the complexity involved is to consider the number of post-translational modifications that membrane proteins may be subjected to prior to attaining their mature state. These include proteolysis, assembly into multimers, glycosylation, addition of a glycophosphate diinositol, glycophosphate inositol GPI anchor, sulfation on tyrosine or carbohydrate moieties, phosphorylation, acylation, prenylation, a list that is undoubtedly not complete. Nevertheless, complete progress has been made. Table 8 summarizes some of the major features of membranes assembly that have emerged till date. So we go to that table, which is already shown to us. Now we're going to the next topic, which is on section A, which is designated as electrical properties of membranes. Now, biological membranes serve as barriers to the passage of ions and polar molecules, a fact that is reflected in their high electrical resistance and capacitance. The electrical resistance is and capacitance. The electrical resistance is usually 10 raised to 3 ohms centimeter raised to minus 2, while the capacitance is 0.5 to 1.5 microfarad microF centimeters raised to minus 2. The corresponding values for artificial membranes are 10 raised to 7 ohms centimeters raised to minus 2, and 0.6 to 0.9 microfarad centimeters raised to minus 2. The lower resistance of biological membranes must result from the presence of proteins and the other ion-carrying substance or of pores in the membranes. The capacitance values for the two types of membranes are very close to those expected for a bilayer with a thickness of Schwaber 2.5 nanometers and a dielectric constant of 2. The electrical potential gradient is steep. Outer cell surfaces usually carry a net negative charge. The result of phosphate groups, of phospholipids, of carboxylate groups and proteins, and the sialic acids attached to glycoproteins. This negatively charged surface layer attracts ions of the opposite charge, counter ions, including protons, and repels those of the same charge. The result is development of a diffuse electrical double layer consisting of the fixed negative charges on the surface and a positive ionic atmosphere extending into the solution for a distance that depends upon the ionic strength. This ionic atmosphere is analogous to that postulated by Debye-Huckel's theory. At the physiological ionic strength of 0.145 m, the thickness of the double layer, taken as the distance at which the electrical potential falls to a certain fraction of that of the cell surface, is about 0.8 nanometers. However, the double layer thickness increases to about 3 times this value at an ionic strength of 10 raised to minus 3 m and to still greater distances at lower ionic strengths. The net surface charge of a cell and the associated electrical double layer are important in interactions between cells and may influence the development of extracellular structures such as basement membrane. The net negative charge on cells also gives rise to an experimentally measurable electrophoretic mobility. A characteristic of living cells is the maintenance of ionic radiance across the plasma membrane, as almost all cells accumulate K+, even pumping it from very dilute external solutions. Cells also exclude sodium, pumping it out from the cytoplasm. If a microelectrode is inserted through a cell membrane and the potential difference is measured between the inside and outside of the cell, a resting potential, which in nerve cells may be as high as 90 MV, is observed. The origin of the potential appears to lie in the concentration of differences of ions from the value of delta G for dilution of an ion, equation 1, and the relationship between Ag delta G and the electric potential, equation 2. The enhanced equation, equation 3, can be derived. All right. And the delta G dilution from A1 to A2 is equal to RT base logarithm A2 by A1. When electrons flow in the external circuit, the Maximum amount of work that can be done per mole of electrochemical reaction minus delta G is given by equation 2. Delta G is equal to NEF is equal to NE into 96.487 kilojoule moles raised to minus 1 into V raised to minus 1 would be equal to minus NE into 23.061 kilocalories mole raised to minus 1 V raised to minus 1 is equation 2. Now the final would be EM is equal to RT by NF logarithm C1 by C2 is equal to 0.5059 by N log C1 by C2 at 25 degrees centigrade. According to this equation, which applies to a single ion for which the membrane is permeable, a tenfold concentration difference across the membrane for a monovalent ion N equal to 1 would lead to a 59 MV membrane potential EM. Since membranes are relatively impermeable to sodium ions, it is generally conceded that for many membranes, the origin of the membrane potential lies mainly with the potassium ion concentration difference, which is maintained by the sodium-potassium ATPase, a more complete equation that takes account of K plus potassium, sodium, and chlorine together with their respective permeabilities. Note that equation 4 is also often called the Nernst equation. Protons are also pumped across cytoplasmic and inner mitochondrial membranes. That is the fourth equation. So if N is equal to 2, E is equal to E0 plus 0.0296 log AH squared by AH2 volts at 25 degrees centigrade. The flow of protons from inside to outside also contributes to the membrane potential. The positive charges of H+, potassium and other cations associated with the external membrane surface are balanced by the negative charges of protein molecules as well as chlorine and phosphate anions that are in or near to the inner surface of the membranes. Another possibility for proton flow has intrigued biophysicists for years. Membranes often display a substantial electrical conductivity in a lateral direction along the membrane surface. Electrical conduction may involve movement of protons along hydrogen bonded lines, example involving ethanolamine, het groups or phosphate groups in bridging water as previously discussed. Alternatively, conduction may depend upon membrane-associated proteins. This lateral proton conduction may be important to many proton-driven membrane processes such as rotation or bacterial flagella, ATP synthase and pumping of ions. And moving on to the next section, B, under the topic of structural organization and function of intracellular organelles. Cells are the structural and functional units of life, as we all know. The smallest organisms are composed of a single cell, while the largest are made up of billions of cells. Even when comparing the most diverse and complex organisms at the cellular level, they are remarkably similar. Even though the human body has over 100 different cell types, they all share certain features that they even have many characteristics in common with plants. From prokaryotes to eukaryotes, it is thought that all organisms living now on Earth derive from a single primordial cell born more than 3 billion years ago. This cell, out-reproducing its competitors, took the lead in the process of cell division, an evolution that eventually covered the Earth with green, changed the composition of its atmosphere, and made it the home of intelligent life. The family resemblances among all organisms seem too strong to be explained in any other way. One important landmark along this evolutionary road occurred about 1.5 billion years ago, when there was a transition from small cells with relatively simple internal structures, the so-called prokaryotic cells, which include the various types of bacteria, to a flourishing of larger and radically more complex eukaryotic cells, such as are found in higher animals and plants. Prokaryotic cells are structurally simple but biochemically diverse. Bacteria are the simplest organisms found in most natural environments. They are spherical or rod-shaped cells, commonly several micrometers in linear dimension. They often possess a tough protective coat called the cell wall, beneath which a plasma membrane encloses a single cytoplasmic compartment containing DNA, RNA proteins, and small molecules. In the electron microscope, this cell interior appears as a matrix of varying texture without any obvious organized internal structure. Bacteria are small and can replicate quickly, simply dividing in two by binary fission. When food is plentiful, survival of the fittest generally means survival of those that can divide the fastest. Under optimal conditions, a single prokaryotic cell can divide every 20 minutes and thereby give rise to 5 billion cells, approximately equal to the present human population on Earth, in less than 11 hours. The ability to divide quickly enables population of bacteria to adapt rapidly to changes in the environment under laboratory conditions. For example, a population of bacteria maintained in a large vat will evolve within a few weeks by spontaneous mutation and natural selection to utilize new types of sugar molecules as carbon sources. In nature, bacteria live in an enormous variety of ecological niches, and they show a corresponding richness in their underlying biochemical composition. Two distantly related groups can be recognized: the eubacteria, which are the commonly encountered forms that inhabit soil, water, and larger living organisms, and the archaebacteria, which are found in such incommodious environments as bogs, ocean depths, salt brines, and hot acid springs. There are species of bacteria that can utilize virtually any type of organic molecule as food, including sugars, amino acids, fats, hydrocarbons, polypeptides, and polysaccharides. Some are even able to obtain their carbon atoms from CO2 and their nitrogen atoms from N2. Despite their relative simplicity, bacteria have existed for longer than any other organisms and still are the most abundant type of cell on Earth. Eukaryotic cells contain several distinctive organelles. As molecular oxygen accumulated in the atmosphere, what happened to the remaining anaerobic organisms with which life had begun? In a world that was rich in oxygen, which they could not use, they were at a severe disadvantage. Some no doubt became extinct, some others either developed a capacity for respiration or found niches in which organism was largely absent, where they could continue as an anaerobic way of life. Others became predators or parasites in aerobic cells, and some, it seems, hit upon a strategy for survival more cunning and vastly richer in implications for the future. They are believed to have they are believed to have formed an intimate association with an aerobic type of cell, living with it in symbiosis. This is the most plausible explanation for the metabolic organization of present-day cells of the eukaryotic type with which this book will be chiefly concerned. Eukaryotic cells, by definition and in contrast to prokaryotic cells, have a nucleus karyon in Greek, which contains most of the cell's DNA, enclosed by a double-layer membrane. The DNA is thereby kept in a compartment separate from the rest of the contents of the cell, the cytoplasm, where most of the cell's metabolic reactions occur. In the cytoplasm, moreover, many distinctive organelles can be recognized. Prominent among these are two types of small bodies, the chloroplasts and the mitochondrion. Each of these is enclosed in its double layer of membrane, which is chemically different from the membrane surrounding the nucleus. Mitochondria are an almost universal feature of eukaryotic cells, whereas chloroplasts are found only in those eukaryotic cells that are capable of photosynthesis, that is, in plants but not in animals or fungi. Both organelles almost certainly have a symbiotic origin. We move on to B1, which is about the cell wall. A cell wall is a fairly rigid layer surrounding a cell, located external to the cell membrane, which provides the cell with structural support, protection, and acts as a filtering mechanism. The cell wall also prevents overexpansion when water enters the cell. They are found in plants, bacteria, fungi, algae, and some archaea. Animals and protozoa do not have cell walls. The materials in a cell wall varies between species. In plants, the strongest component of the complex cell wall is a carbohydrate polymer called cellulose. In bacteria, peptidoglycan forms the cell wall. Archaean cell walls have various compositions and may be formed of glycoprotein S layers, pseudopeptidoglycan, or polysaccharides. Fungi possess cell walls of chitin, and algae typically possesses walls constructed of glycoproteins and polysaccharides. However, certain algal species may have a cell wall composed of silicic acid. Often, other accessory molecules are found anchored to the cell wall. Properties. The cell wall serves a similar purpose in those organisms that possess them. The wall gives cells rigidity and strength, offering protection against mechanical stress. In multicellular organisms, it permits the organism to build and hold its shape, morphogenesis. The cell wall also limits the entry of large molecules that may be toxic to the cell. It further permits the creation of a stable osmotic environment by preventing osmotic lysis and helping to retain water. The composition properties and form of the cell wall may change during the cell cycle and depend on growth conditions. Rigidity. In most cells, the cell wall is semi-rigid, meaning that it will bend somewhat rather than hold in a fixed shape. This flexibility is seen when plants wilt so that their stems and leaves begin to droop, or in seaweeds that bend in water currents. Wall rigidity seen in healthy plants results from a combination of the wall construction and turgor pressure. The rigidity of the cell wall thus results in part from inflation of the cell contained. This inflation is a result of the passive uptake of water. Other cell walls are inflexible. In plants, a secondary cell wall is a thicker additional layer of cellulose. Additional layers may be formed containing lignin in xylem cell walls or containing suberin in cork cell walls. These compounds are rigid and waterproof, making the secondary wall stiff. Both wood and bark shells of trees have secondary walls. Other parts of plants, such as the leaf stalk, may acquire similar reinforcement to resist the strain of physical forces. Certain single-cell protists and algae also produce a rigid wall. Diatoms build a frost tube from silica extracted from the surrounding water. Radiolarians also produce a red test from minerals. Many green algae, such as the dacylcladiales, encase their cells in a secreted skeleton of calcium carbonate. In each case, the wall is rigid and essentially inorganic. Permeability. The primary cell wall of most plants is semi-permeable and permit the passage of small molecules and small proteins with size exclusion estimated to be 30 to 60 kDa. Key nutrients, especially water and carbon dioxide, are distributed throughout the plant from cell wall to cell wall in hypoplastic flow. Plant cell walls. Composition. The major carbohydrates making up the primary growing cell wall are cellulose, hemicellulose, and pectin. The cellulose microfibers are linked via hemicellulose tethers to form the cellulose-hemicellulose network, which is embedded in the pectin matrix. The most common hemicellulose in the primary cell wall is xyloglucan. In grass cell walls, xyloglucan and pectin are reduced in abundance and partially replaced by glucurono-arabinoxylin as hemicellulose. Primary cell walls characteristically grow by a mechanism called acid growth, which involves turgor-driven movement of the strong cellulose microfibers within the weaker hemicellulose pectin matrix, catalyzed by expansion proteins. The major polymer that makes up wood, lastly secondary walls, include cellulose 35 to 50%, xylem, a type of hemicellulose 20 to 35%, and a complex phenolic polymer called lignin, 10 to 25%. Lignin penetrates the spaces in the cell wall between cellulose, hemicellulose, and pectin components, driving out water and strengthening the wall. Secondary walls, especially in grasses, may also contain microscopic silicate crystals, which may strengthen the wall and protect it from herbivores. Plant cells also contain numerous enzymes such as hydrolyzes. We're continuing from where we stopped, the formation of the cell wall. The middle lamina is laid down first, formed from the cell plate during cytokinesis, and the primary cell wall is then deposited inside the middle lamella. The actual structure of the cell wall is not clearly defined, and several models exist. The covalently linked cross model, the tether model, the diffuse layer model, and the stratified layer model. However, the primary cell wall can be defined as a composed of cellulose microfibrils aligned at all angles. Microfibrils are held together by hydrogen bonds to provide a high tensile strength. Excuse me. The cell walls are held together and share the gelatinous membrane called the middle lamella, which contains magnesium and calcium pectate, salts of pectic acid. Cells interact, though, through plasmodesmata, which are the interconnecting channels of cytoplasm that connect to the protoplasm adjacent to cells across the cell wall. In some plants and cell types, after maximum size or point in development has been reached, a secondary wall is constructed between the plant cell and the primary wall. Unlike the primary wall, the microfibrils are aligned mostly in the same direction, and with each additional layer, the orientation changes slightly. Cells with secondary cell walls are rigid. Cell-to-cell communication is possible through pits in the secondary cell wall that allow plasmodesma to connect cells through the secondary cell walls. Trees modify cell walls in their branches to reinforce and support structure. Conifers, such as pine, produce thicker cell walls on the underside of branches to push their branches upwards. The resulting wood is called compression wood. By contrast, hardwood trees reinforce the walls on the upper side of branches to pull their branches up. This is known as tension wood. Additional thickening may occur in other parts of the plant in response to mechanical stress. Algal cell walls. Like plants, algae have cell walls. Algal cell walls contain cellulose and a variety of glycoproteins. The inclusion of additional polysaccharides in algal cell walls is used as a feature for algal taxonomy. Monosyl from microfibrils in the cell walls of a number of marine green algae, including those from the genera Codium, Dasycladus, and Acetabularia, as well as in the walls of some red algae like Porphyra and Bangla. Xylanes, alginic acid, is a common polysaccharide in the cell walls of brown algae. Sulfonated polysaccharides occur in the cell walls of most algae. Those common in red algae include agarose, carrageenan, porphyrin, fusilarin, and phonorin. Other compounds that may accumulate in algal cell walls include sporopollen and calcium ions. The group of algae known as the diatoms synthesize their cell walls, also known as prostoles or valves, from silicic acid, specifically orthosilicic acid, H4SiO4. The acid is polymerized intracellularly. Then the wall is extruded to protect the cell, significantly relative to the organic cell walls produced by other groups. Silica prostoles require less energy to synthesize, approximately 8%, potentially a major saving on the overall cell energy budget, and possibly an explanation for higher growth rates in diatoms. Fungal cell walls. There are several groups of organisms that may be called fungi or fungi. Some of these groups have been transferred out of the kingdom fungi, in part because of fundamental biochemical differences in their composition of the cell wall. Most true fungi have a cell wall consisting largely of chitin and other polysaccharides. The true fungi do not have cellulose in their cell walls, but some fungus-like organisms do. True fungi. Not all species of fungi have cell walls, but in those that do, the plasma membrane is followed by 3 layers of cell wall material. From inside out, these are a chitin layer polymer consisting mainly of unbranched chains of N-acetyl D-glucosamine, a layer of beta-1,3-glucan, a layer of manoproteins, manos containing glycoproteins which are heavily glycosylated at the outset of the cell. The fungus-like protists, the group Oomycetes, also known as water moles, as saprotrophic plant pathogens like fungi, and recently they were widely believed to be fungi, but structurally and molecular evidence has led to their reclassification as heteroconchs, related to autotrophic brown algae and diatoms. Unlike fungi, Oomycetes typically possess cell walls of cellulose and glucans rather than chitin, although some genera such as Echyla, Eclea, and Saprolegnia do have chitin in their walls. The fraction of cellulose in the walls is no more than 4 to 20%. far less than the fraction comprised by glucans. Oomycete cell wall can also contain the amino acid hydroxyproline, which is not found in fungal cell walls. The dictyostellates are another group formerly classified among the fungi. They are similar molds. They are slime molds that feed as unicellular amoebae, but aggregate into a reproductive stalk and sporangium under certain conditions. Cells of the reproductive stalk as well as the spores formed at the apex possess a cellulose wall. The spore wall has been shown to possess 3 layers, the middle of which is composed primarily of cellulose, and the innermost is sensitive to cellulase and tronase. Prokaryotic cell walls, bacterial cell walls. Around the outside of the cell wall is a bacterial cell wall. Bacterial cell walls are made up of peptidoglycans, also called murine, which is made from polysaccharide chains cross-linked by unusual peptides containing D amino acids. Bacterial cell walls are different from the cell walls of plants and fungi, which are made of cellulose and chitin respectively. The cell wall of bacteria is also distinct from that of archaea, which do not contain peptidoglycan. The cell wall is essential to the survival of many bacteria. The antibiotic penicillin is able to kill bacteria by inhibiting a step in the synthesis of peptidoglycan. There are broadly speaking 2 different types of cell wall in bacteria called the gram-positive and gram-negative. Their names originate from the reaction of cell walls, cells to the gram stain, a test long employed for the classification of bacterial species. Gram-positive bacteria possess a thick cell wall containing many layers of peptidoglycan and teichoic acids. In contrast, gram-negative bacteria have a relatively thin cell wall consisting of a few layers of peptidoglycans surrounded by a second lipid membrane containing lipopolysaccharides and lipoproteins. Most bacteria have the gram-negative cell wall, and only the firmicutes and actinobacteria, previously known as low G+C and high G+C gram-positive bacteria respectively, have the alternative gram-positive arrangement. These differences in structure can produce differences in antibiotic susceptibility. For instance, vancomycin can only gram-positive bacteria and is ineffective against gram-negative pathogens, such as Haemophilus influenza or Pseudomonas aeruginosa. And to the diagram, this is a simplified schematic cell wall in a gram-positive bacterium, showing a plasma membrane, teichoic acid, not shown, archaeal cell walls, although not truly unique. The cell walls of archaea are unusual, whereas peptidoglycan is a standard component of all bacterial cell walls. All archaeal cell wall lack peptidoglycan, with the exception of one group of methanogens. In that group, the peptidoglycan is a modified form very different from the kind found in bacteria. There are 4 types of cell wall currently known among the archaea. One type of archaeal cell wall is that composed of pseudopeptidoglycan, also called pseudomurine. This type of wall is found in some methanogens, such as Methanobacterium and Methanothermus. While the overall structure of archaeal pseudopeptidoglycan superficially resembles that of bacterial peptidoglycan, there are a number of significant chemical differences. Like the peptidoglycan found in bacterial cell walls, peptidoglycanosis consists of polymer chains of glycan crossed, linked by short peptide connections. However, unlike peptidoglycan, the sugar N-acetylmuramic acid is replaced by N-acetyl-allosaminuronic acid, and the 2 sugars are bonded with beta-1,3 glycosidic linkage instead of beta-1,4. Additionally, the cross-linking peptides are L-amino acids rather than D-amino acids, as they are in bacteria. A second type of archaeal cell wall is found in Methanosarcina and Halococcus. This type of cell wall is composed entirely of a thick layer of polysaccharides, which may be sulfated in the case of Halococcus. Structure in this type of wall is complex and is yet not so fully investigated. A third type of wall among the archaea consists of glycoprotein and occurs in the hyperthermophiles. Halobacterium and some methanogens in halobacterium. The proteins in this cell wall have a high content of acidic amino acids, giving the wall an overall negative charge. The result is an unstable structure that is stabilized by the presence of large quantities of positive sodium that neutralizes the charge. Consequently, halobacterium thrives only under conditions with high stability. In other archaea, such as methanomicrobium and D-sulfurococcus, the wall may be composed only of surface layer proteins known as S-layer. S-layers are common in bacteria, where they serve as either the sole cell wall component or an outer layer in the conjunction with peptidoglycan and murine. Most archaeas are